So who has questions? Cool. That's good. Can you guys hear me? Are we here? Let's try it this way. Okay, we don't currently have questions. So if you've been at or seen the previous reviews, then I think you know that I don't have anything additional prepared. I'm just here to go over any concepts. There we go. Okay. Okay, are the neuron, oh yeah, so the labeling a neuron on the practice quiz was, for some reason, the key was totally wrong. So you probably didn't get them all wrong. You probably got them right. I was able to fix it. So if you go back and look at it now, you'll see, hopefully, that they're right. I have no idea how it got like that, because I thought that I put it all in there properly. So yeah, so that's corrected. The diagram should be correct now. All right, so the resting membrane. So can you be more specific on what your question is in regards to resting membrane potential? Okay, let me pull that up. Okay, so resting membrane potential is definitely an important concept, since it's basically the basis upon which we can have electrical activity. So for resting membrane potential, the most important thing to understand is that the cell has these sodium-potassium pumps. So the cell is using ATP. This is an active transport process, right? So primary active transport. And it's pumping sodium out of the cell and potassium in because we want lots and lots of sodium in the cell and lots of potassium in the cell and lots of sodium outside of the cell. And then there's always going to be more sodiums getting pumped out than potassiums get pumped in. And there's typically these little always open leak channels allowing a little bit of potassium to trickle back out. And the net result of that is that there's always more positive charge outside of the cell and negative charge inside of the cell. And that is our resting membrane potential. So this negative charge, and you don't need to know the number, but it's typically about negative 70 millivolts, is being maintained by this active transport system that is basically constantly running because the cell isn't usually resting for very long. So it's always having to reset because it's really hard to maintain these very high concentration gradients. We have so much sodium out here, lots and lots and lots and lots of sodium, and so much potassium in here. And anytime the cell does anything, it's going to let a little bit of sodium in, and it's going to let a little bit of potassium out, and then it's got to reset the whole thing every single time. So when we talk about our resting membrane potential, we're talking about this actively maintained state of a high potassium inside of the cell with negative charge and high sodium outside of the cell. Do we have any other questions in regards to resting membrane potential? Okay. What else do we have questions about? All right, stimulation of a neuron. Go back to that then. So when we talk about stimulation of neurons, there's kind of a lot of different ways to approach it. So the basic way to look at it is that neurons are typically stimulated at synapses. The other way they get stimulated is by being sensory neurons and getting sensory stimulation. So can you specify which one you have questions about, Sandra? So there's different aspects to stimulating the neuron. So are you referring to stimulation across a synapse, or are you referring to sensory neurons being stimulated by whatever it is they respond to? Okay, that's different. Okay, pretty sure that's in here. All right, so when we're looking at things that stimulate sensory neurons, it's going to be a little different from what's happening across a synapse. So the thing that's happening here is that the neuron is basically prepped to respond to some sort of sensory modality, right? So temperature, pain, chemical changes, pressure, anything like that. And when we look at how they're actually responding, generally speaking, what happens is that there's going to be some sort of gated ion channels in the place on the neuron that is being stimulated. Okay, now we really didn't go into this. We talked about the types of things that can stimulate them. We talked about, you know, like how those signals can be modified, but we didn't go into this in a lot of detail. Okay, but what's going to happen here is, you know, let's say this guy responds to temperature, and so whenever these ion channels get warm enough, it's going to open gated channels, and that's going to allow a little bit of sodium in. And that sodium is going to partially depolarize the membrane by generating graded potentials. And if it generates enough of it to depolarize this to threshold, then we're going to trigger an action potential, and that action potential is going to travel down the entire length of the sensory neuron to the synapse on the other side. Okay, so the way that this works for the sensory neurons is very similar to the way it works for any neuron. It's just that instead of a synapse here, where this is the postsynaptic neuron, it's sensory channels that can be opened at the level of the dendrites that can then trigger an action potential, and then it'll synapse with the next neuron down the line, heading up to the brain typically. But we didn't get into the details of this because it's beyond the level of the class. So when we talked about graded potentials in the first lecture, what we were talking about is the kinds of things that would be triggering or stimulating a sensory neuron. So these graded potentials are the ones that are going to occur at the level of the dendrites. That's not what I want. Oh, that works too. At the level of the dendrites, and we're, sorry, it's always something. Stop it. And we're stimulating, again, these gated ion channels The more stimulation, the larger this graded potential, right? So remember, graded potentials will be stronger with a stronger stimulus. And as long as we hit threshold, we'll trigger an action potential throughout the entire neuron. So how we're getting that stimulus, triggering it, this is all we need to know about that. It's just opening a gated ion channel. So did you have any more specific questions about that? Okay, good. All right, so what else are we feeling confused about? Okay, you're gonna have to be more specific. So you have a question about the cranial nerves. What is your question though? I really need you guys to ask questions and not just state things that we learned. Richard, can you flesh out your question so I understand exactly where you need more information? Okay, so are those nerves that you want me to go back over? Are you not understanding their functions? Where's the actual issue here? Let's see. Where did I put those? Okay, well, let's try that again then. All right, so this is the glossopharyngeal. This is the one where I have this typo that apparently I will never fix on my slide because it's cranial nerve 9. Okay. So the glossopharyngeal has kind of a variety of complicated functions, but for the most part, its job is to help control the muscles in the tongue and the throat that aid in swallowing. It also controls one of our salivary gland pairs. So we've got these salivary glands back here called the parotids that it controls when that's secreting saliva. And then it's also anything that you can taste on the back of your tongue. So the posterior 1/3, all right? Here's your tongue. Here's the back third. Anything you taste there is being carried by this guy. So he's kind of got a mixed bag of the back of your mouth stuff, essentially, that that it's got the axons for those guys. Okay, so that's what the glossopharyngeal does, and that's why it's named that too, because remember that glosso refers to tongue, and pharynx is basically your throat. So because it's got to do with things in your tongue and throat, it was just named descriptively for that. And then the accessory nerve, or cranial nerve 11, is actually just controlling 2 muscles in your neck. So the trapezius muscle is that muscle at the back of your neck. So you know how people have, when they work out, especially guys that work out a lot, and they get that like stacked look to their top of their shoulders? That's the trapezius muscle. And then the sternocleidomastoid muscle is the one that runs from the front of your neck around to the base of your skull. So right here. And literally that's what this nerve does. It tells those muscles when they're supposed to be contracting. So, and it doesn't do much, but you know, those muscles are kind of helpful in letting you move your neck around and stuff. So, you know, we still like it. And that's pretty much how those two work. So when we talk about these nerves, what we're looking at is what do they control? What is their function? And that's really all you need to know about them. There's of course a lot more information to know about them, but we don't need to know it right now. All right, while I'm here, does anybody else have cranial nerve questions? Something they didn't understand about innervation? I think that's a no to the cranial nerves. All right, hormones. That one. So again, this is something where we didn't really go over the details. We don't really need to know the nitty gritty. Okay, so basically, a variety of things can happen when a hormone binds to a target cell, and it's just based on how that receptor is set up. And so things that often happen when a hormone binds are these. These are kind of our options for responses. And so this is essentially as much as we need to know on how these hormones work. Now, if you read the book went into it in a little bit more detail, but you don't need to know that. I didn't cover it because it's too much detail for the level that we're learning it. So if you read the book and you're not sure how like G proteins work or something like that and second messengers, you don't have to. I'm not going to ask you about it. I didn't cover it. Okay. So the receptors can be ion channels. So they can like literally just be ligand gated ion channels. But a lot of them control the inner workings of the cell in different ways. So they can actually influence what proteins that the cell is making. They can influence whether those proteins, especially as enzymes, are actually functional or not. They can tell the cell to secrete something, and we see that especially with those tropic hormones. So remember, the tropic hormones are the ones that stimulate cells to other endocrine organs to release hormones. So that's a classic example of inducing secretory activity. And then when we have these guys that are related to like growth and development, sometimes they're stimulating mitosis. So we're encouraging cells in the cell cycle. So things like growth hormone, insulin-like growth factor, and thyroid hormone are potentially going to be stimulating some types of cells in the cell cycle, for example. But how they're doing it, the details of what happens when a hormone binds to its receptor, that's beyond what we need to know. So really the only other detail that you need to know is that the water-soluble ones, they're binding to receptors that are proteins in the membrane. And then the lipid-soluble or water-insoluble ones, they're actually able to enter the cell because they're lipid-soluble, so they can freely cross the plasma membrane. And so the receptors are inside the cell somewhere, and they bind internally instead. And that's it. That's as far as we went as far as the mechanisms of action. So unless I misunderstood the question, in which case just let me know what you were referring to. So the ion channels, as far as like what happens during an action potential, is that what you mean? Right, hold on. Let me look at that real quick. I think I was just asking about them, but Okay, yep. Okay, so yeah, we can go over that. And I know they're called quizzes, guys, but they're really just meant to replace the worksheets we were doing. So they really aren't quizzes. Um, just to clarify that, because I don't know. I mean, if it helps you to think of it as a quiz, you can. Okay, so that's going to take us back here. Okay, so we obviously deal with a lot of different channels, and so let's kind of take a look at the role of each of them in the electrical activity. So we talked about the sodium potassium pump already. Ligand-gated ion channels are basically going to be neurotransmitter receptors. And so their role is to create graded potentials in postsynaptic neurons. When we look at mechanically gated ion channels, they're only going to be in sensory neurons who, for the most part, are mechanoreceptors. So remember that we have different classes of sensory neurons based on the stimulus modality. And so when they are stimulated by something mechanical or physical, then that's where we're going to find these guys, because this is how they're able to respond to that, is with these mechanically gated ion channels. As far as voltage gated ion channels, there are three kinds. So there's the sodium, the potassium, and the calcium. So sodium gated channels are the ones that open to depolarize the cell during an action potential. Potassium repolarize the cell during an action potential. And then the calcium gated channels are actually the ones that trigger the release of neurotransmitter in the presynaptic neuron. So the end result of these guys continuing an action potential all the way down an axon is to open voltage-gated calcium channels, which then calcium floods into the cell, it releases neurotransmitter across the synapse, and then we're transmitting the signal from a presynaptic neuron to a postsynaptic neuron. All right, did I miss any of those before we go to the next question? Okay, so the negative and positive feedback hormones. So there is literally only one hormone that we see with a positive feedback effect, and that's going to be oxytocin. So we're not really dealing with the positive feedback loops in the endocrine system. I mean, I didn't bring it up, we didn't talk about it. We'll address it when we get to the reproductive system and we look at childbirth. So every loop that we talked about in the endocrine lecture is a negative feedback loop. All right, is there anything else? There must be something. So the exam layout, yes. So it's going to be just like the last one, in that it'll be 40 multiple choice questions, randomly selected from test banks, based on all the material that we covered. And then you'll have 5 short answer questions. You'll have 2 hours to complete the whole thing. You'll see each question once and you won't be able to go back to it. And then yeah, and then once you start taking the exam, you have to finish it, but you can access it at any time on Friday or Saturday is when it'll be available. So a couple of words on that too, as I was grading your last one. I think it's important to emphasize that you need to make sure you're answering the question that's being asked, not the question you think is being asked. Because I saw a lot of responses that were addressing what I would call a key word in the question, as opposed to actually answering the question. I also saw, and some of you probably already know this if you looked at your exams after I graded them, but I also saw a lot of cheating on the exam. And if you took this exam without looking any answers up, then I commend you for your honesty. And for the rest of you, if I catch you cheating, I can't give you points for it. I know it's tempting. There's nobody there to police you. Nobody necessarily is going to know, except that I can tell a lot of the time. And if I can prove it, then I'm not giving you points. And so I saw lots of evidence of people were clearly Googling answers. And not only that, not even answering the question being asked. So on top of the cheating, you were still getting it wrong regardless. It's not acceptable. I understand that we're all in a weird position and the opportunity is there, and I can't technically stop you from doing it. But I can fail you. So please resist the temptation to look up answers. I set this up so you guys have lots of time to study. I give you time on the exam to think about what's going on. And I guarantee you that there will not be a single short answer question that can be answered in a list going forward. They're all going to be, how does this work and why does this happen? And you're not going to be able to Google the answers. And if you try, I'm going to know, and you're going to get a 0 on the entire exam. Okay? So if you're not doing that, then excellent. You've got nothing to worry about. If that's something that you were pursuing as a testing strategy, maybe stop doing it. Okay? That was a policy in the syllabus. That is the policy of every single school that exists. Cheating is not tolerated. And if I can prove that you did it, then I will be emailing the relevant authorities at the school so that they're aware that this is something you're pursuing. Okay? If you can't tell, it really pisses me off. So you've got that on top of it. And I'll be sending an announcement explaining all that as well in case people aren't watching this review, which I don't blame them for. They don't want to. Any questions? Any other questions about the material, confusions, clarifications, concepts that you want gone over again. If you mean like the parts of the brain that we went over, like the cerebral cortex and the basal nuclei and like thalamus and stuff like that, then yes. So if I went over it and kind of gave you a basic functions of this part of the brain, you should know it. Where they're located, that's a good question. The No, not really. So the not beyond like what I put on the worksheet is probably the extent. I don't expect you to know like all the details because that's something we would cover in lab. So, I mean, I should be able to put up a brain and be like, hey, is this the cerebellum? And you're like, oh yeah, that's the cerebellum, but I wouldn't expect you to know anything beyond that. Make sure you understand your spinal cord anatomy though, because I did go over that. So I've got the slides explaining all the different parts and stuff. So you should know that because I went into the details on that, or not the details, but the overview. So the grading, there definitely are a little bit of changes, and unfortunately, it's harder for you guys to track your grades now because until I actually put the exams in, they don't show up on your grade book. So it's kind of in flux, and I appreciate that that's not ideal for you, but it's the best I can do under the circumstances. These worksheets are counting towards the participation points because that's all I have since we're not doing attendance, and otherwise it's basically just the exams. But I'm pretty sure I added an exam. I think there's one more exam than there was before, so that does hopefully help spread out the points a little bit. Yeah. So, yeah, adding exams is hard to do in live classes because we just only have so many days of class, but this way it's tests on fewer lectures, so there's less material to study, and then you have a test before your final exam as well. all the information. I mean, you need to know all of them. The only one you don't actually need to know is the melanocyte stimulating hormone, which I just kind of throw in for posterity. But yes, you should know all the information. I pulled out anything that you don't need to know. Now, I will say if there's something on a diagram that I did not write down on a slide, you don't need to know it. So the diagrams do contain more detail than you need to know. So if I expect you to know it, then I wrote it on the slides. So I would prefer not to sit here for the next half an hour in complete silence. If you guys have questions, please just ask them. If not, we can all stop staring at the screen. But I don't want to cut anybody off. Okay, learning objectives. So yeah, ideally, if you're able to answer the learning objectives for the lectures, then you should be able to do well on the exam. That's kind of the goal here. And that's where I'm going to be drawing all your short answer questions from as well. So definitely use them to guide your learning because they are distilling what we're covering as like concrete points. Okay, so There is an endocrine worksheet up too now, and I put the answers in that one. So you can work through that as well to make sure you're kind of getting all your endocrine stuff covered. But yeah, if you're looking at the learning objectives and then trying to answer them based on the lecture material, but I definitely don't recommend just rereading the lectures. It's a very inefficient way to study. So make sure that you're pulling the material from them and actually committing yourself to knowing it without looking at it, okay? And you can still email me if you have any questions before the tests. And yeah, we're not done yet. We've got more to go. So we'll try to learn from this round and improve it on the next one. Okay. So thanks for coming. Thanks for the questions. And I will see you all next week. Stop recording.